experiments in cell biology, a video. Biologists frequently propose models to explain phenomena that they are studying. Often, conflicting models can fit the same set of results. And so then biologists must try to devise experiments that will distinguish between conflicting models. This video will present such a situation. IRE1 is a key element in the pathway connecting the accumulation of unfolded proteins in the lumen of the endoplasmic reticulum, or ER, leading to the induction of the unfolded protein response in all eukaryotic cells. In this video, we will discuss two possible models for the mechanism of activation of the unfolded protein response via IRE1, an experimental attempt to distinguish between those models, and the sadly ambiguous interpretation of those experiments. IRE1 is a transmembrane protein. It looks a lot like plasmamembrane receptors, but IRE1 is found in the membrane of the endoplasmic reticulum. The domain of IRE1 in the ER lumen appears to be largely disordered, like an unfolded protein. The domain in the membrane itself has the hydrophobic sequence of a typical intramembrane domain for a transmembrane protein. And the cytoplasmic domain of IRE1 has two identifiable sequence and structural motifs, one that predicts a kinase domain and another that predicts a ribonuclease domain. Experiments demonstrate that the kinase domain of one IRE1 molecule can phosphorylate the cytoplasmic domain of another IRE1 molecule. Such a reaction is a transphosphorylation. A series of experiments shows that transphosphorylation can activate the IRE1 ribonuclease domain, and that in turn is important for events downstream of IRE1 in the UPR pathway. In yeast, for example, we know that the substrate for this ribonuclease activity is HAC1's messenger RNA, and this cleavage is the first step into turning that message into its translatable form, translation that in turn leads to activation of the transcriptional responses of the UPR. So what events happen inside the lumen of the ER that are necessary and sufficient for the kinase of one IRE1 molecule to phosphorylate another IRE1 molecule, and so turn on the ribonuclease activity of IRE1? An important piece of evidence in answer to this question came from an experiment that showed that overexpression of IRE1 is sufficient to drive transphosphorylation and thus activation of the ribonuclease activity, even in the absence of accumulating unfolded proteins that are the normal trigger of the UPR. This result strongly indicates that self-association of IRE1 molecules, that is, interactions between IRE1 molecules, driven in this experiment by overexpression of the protein, is sufficient to turn on the UPR. Well, that's neat. But we know from other experiments that in cells accumulating unfolded proteins, induction of the UPR is not caused by overexpression of IRE1. That is, defects in processing of newly synthesized proteins imported into the ER lumen, defects that lead to the accumulation of unfolded proteins, do not cause overexpression of IRE1 molecules. So overexpression is sufficient, but not necessary. We are interested in understanding how might the activation of the kinase domain of IRE1 happen under physiological conditions with normal levels of IRE1 protein. The nature of the molecular interactions we know about upstream of IRE1 activation suggests some possible mechanisms. The BIP protein is involved in one of those interesting molecular interactions upstream of IRE1. BIP acts as a chaperone, facilitating the successful folding of newly synthesized proteins in the ER. BIP, like other chaperones, can bind to unfolded proteins and, in an ATP-dependent fashion, dissociate from those proteins in a way that gives them a chance to try to fold again. If they fold successfully, then mission accomplished. So one possibility is that BIP interacts non-covalently with unfolded proteins until the level of those unfolded proteins overwhelms the amount of BIP available in the ER lumen. So in this model, we form a hypothesis that those excess unfolded proteins, which don't have any BIP to bind to, can and will instead bind to the disordered domains of IRE1 in the lumen and bring them into close proximity with each other. Please note that in this scheme, the unfolded proteins are an activator of IRE1 self-association and subsequent transmolecular activation. And in this scheme, BIP acts as an inhibitor of that activator. How can we test this experimentally? Well, we can ask what happens if we overexpress BIP. If this model is correct, more BIP will lead to fewer unbound, unfolded proteins. And that in turn will lead to less self-association of IRE1, and thus less activation of the UPR. And indeed, when researchers conducted this experiment, they learned that if they overexpress BIP, they can indeed avoid triggering the unfolded protein response. So this result is consistent with our first model. But let's consider another possible mechanism. We noted earlier that the luminal domain of IRE1 appears to be highly disordered, just like an unfolded protein. We know that unfolded proteins tend to self-associate as aggregates. And so now we can ask, what if BIP binds to the highly disordered domain of IRE1 in the lumen, just as this chaperone protein binds to other unfolded proteins? Then we would hypothesize that BIP bound to those luminal domains of IRE1 prevents them from binding to one another. So then, as the level of unfolded protein rises, the unfolded proteins can also bind to BIP, thus competing with IRE1 for the amount of BIP that is present. The consequences of this reaction would be that as the unfolded proteins increase, more and more BIP dissociates from IRE1 to bind them, leaving the luminal domains of IRE1 free to self-associate, transphosphorylate, and activate. In this second model, then, BIP inhibits the IRE1 self-association reaction itself, and we would say that the unfolded proteins act to interfere with, that is, to inhibit, that inhibitor. Well, how might we test this model? The model predicts that if we overexpress BIP, we prevent triggering the UPR. Fine, but you'll remember under the first model, in which BIP inhibits the ability of unfolded proteins to activate the UPR, overexpression of BIP has the same phenotype. Therefore, we cannot distinguish between our two models by overexpressing BIP. This situation is one that we see often with respect to biological circuits that the cell regulates, and it presents an investigational problem. Here we know that overexpression of unfolded proteins activates IRE1. We also know that overexpression of BIP inhibits that unfolded protein response. But these two statements do not allow us to rigorously distinguish between the two models. This scenario is an excellent example of the fact that searching for answers in science is not always straightforward. We need to keep designing new experiments to gain more information to support or refute existing models, or that may lead to completely new models. Is there any experimental way out of this dilemma? An experiment that distinguishes between the two models? Think about your answer, and be prepared to share your thoughts on one of those experiments. Below, we posted some questions about this experiment. Please read them through and post your answers to them on the discussion forum. It may be that you'll have questions of your own. Take them to the forum too, so that the instructors and your colleagues can respond to them.